Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Hi students. Our topic today will be on antiviral drugs 2, part 1. In this lecture, we will cover the antiretroviral agents. I'm sure all of you already know what is retrovirus. Retrovirus is a group of viruses that belong to the family of Retroviridae, and they carry their genetic materials in the form of ribonucleic acid or RNA. One of the most popular examples of retrovirus in human is human immunodeficiency virus or HIV. I hope all of you already read my lecture notes. Let us start. These are the learning objectives or learning outcomes of this topic. By the end of this lecture, students are expected to know classification of antiretroviral drugs, goals of antiretroviral therapy, mechanism of action of antiretroviral agents, and the concept of antiretroviral therapy or ART. Before we go to the antiretroviral drugs, let us revise how is the structure of a retrovirus. Variants of retroviruses consist of envelope particles about 100 to 120 nanometer in diameter. It is an icosahedral shape with 20 sides or faces. At the outer structure, there are two groups of glycoprotein spikes, namely glycoprotein 120 and glycoprotein 41, which protrude from the lipid bilayer membrane, or we call it envelope. Inside the envelope, there are matrix protein and capsid. In the capsid, there are two copies of single-strand RNA, which carries the genetic materials of the virus. Apart from that, there are three viral enzymes that are essential for the maturation and replication of the virus, namely integrase, reverse transcriptase, and protease. Integrase, which mediates the integration of viral complementary DNA or cDNA into the host DNA. Reverse transcriptase is the enzyme that catalyzes the transcription of retroviral RNA into cDNA. And protease is the enzyme that cleave or cut the new long chain protein into smaller protein for the maturation of the new virus. These three enzymes are very important for you to know because all of them will be the target of our antiretroviral drugs that will be discussed after this. This diagram summarizes the replication process of HIV virus. It starts with the binding of the virus to the CD4 cell, then it fuses to enter the cell. Next, the virus uncoats its genetics materials, followed by replication process, which catalyzed by the reverse transcriptase enzyme. Next, the virus cDNA will be integrated into the host cDNA by the enzyme integrase. After the translation process, the long chain protein will combine with the new RNAs during the assembly step to form a new virus particle. The final step is the budding process, in which immature non-infectious HIV pushes itself out of the host CD4 cell. This non-infectious HIV cannot infect another CD4 cell yet. Once outside the CD4 cell, the new HIV releases protease. Protease breaks up the long protein chains in the immature virus, creating a mature infectious virus. Before we go further, let us see what are the aims or goals of antiretroviral therapy. First is to improve the quality of life and prolongation of life. Then to reduce HIV-related morbidity and mortality. To achieve reduction in viral load in order to stop or delay disease progress. To restore and preserve the immune function. To minimize side effects of drugs. And finally, to reduce HIV transmission. These are the classification of antiretroviral drugs. As you can see here, there are 5 main groups, namely nucleoside or nucleotide reverse transcriptase inhibitors or NRTIs, non-nucleoside reverse transcriptase inhibitors or NNRTIs, protease inhibitors, integrase inhibitors, and fusion inhibitors. As mentioned before, 4 over 5 of these drugs make the viral enzyme as their target. This diagram summarizes the site of actions of the drugs that I have mentioned before. It starts with the fusion or entry inhibitors, which inhibit the virus from entering the host cell. Then NRTI and NNRTI block the viral replication process. Integrase inhibitors interfere with the integration of virus DNA into the host DNA. And finally, protease inhibitors block the action of viral protease enzyme, which plays important role in cleaving the long-chain protein for the viral maturation process. Okay, that's all for now. We will cover the antiretroviral drugs in detail in my next video. Have a nice day. Assalamualaikum. Bye-bye.